Good afternoon. Good afternoon. Welcome to Dr. B.R. Ambedkar Open University. Yesterday we talked about the neurobiology of the insects, and today we'll be seeing the insecticides that are derived from the plants and organoflorins as well. So let me share the slides. So today we'll be dealing with the botanical insecticides, in which primarily we'll be seeing the chemical structure in relation to toxicity and mode of action of the synthesized pesticides which have been developed based on the plant products. So basically in this chapter, only nicotinoids, pyrethranes, neem and retinoids have been included for the reason that these are the compounds that are not only commercialized, but research on these plants has led to the development of new molecules. And each of these compounds has a unique mode of action. So coming to the nicotine, so it represents a class of compounds, nicotinoids, with a unique mode of action. Its biological activity is due mainly to its interaction with acetylcholine receptor. So it is fairly high mammalian toxicity. It has fairly high mammalian toxicity with acute LD50 values. In the case of mouse, for 24 hours for oral and 16 for subcutaneous and 7 for intravenous administrations. It is a narrow insecticidal spectrum it has, and it has very short lifespan, and it is metabolized and excreted fairly fast in insects. And coming to the structure, you can see the pyridine and pyrrolidine groups and along with the methyl group here. This is a nicotine molecular structure. So two other closely related compounds are nornicotine and anabastin. Anabastin that are present in these plants also have some insecticidal activities. So here you can see nicotine, nornicotine and anabastin. So to the insecticidal activities they possess they mimic the nicotine. It should be structurally similar to nicotine and capable of binding to nicotine acetylcholine receptors. It has high affinity for nicotine acetylcholine receptors. The molecule must bind strongly to insects, leading to overstimulation, and it should disturb the neurotransmission in insects, causing paralysis or death. Preferably, it is more toxic to insects than to mammals or non-target species. And the compound should have a prolonged stay so that it can show its toxic effects. Free nicotine is basically colorless, pale yellow in color, and it is miscible with water below 60 degrees. And coming to lipophilicity, good solubility in lipids is often essential for crossing the insect cell membrane. So these are certain criteria that need these compounds to be more insecticidal effective. And it should be chemically stable to withstand environmental conditions. The molecule must be readily absorbed through the insect's cuticle, and it should ideally break down into non-toxic metabolites in the environment. And it's often effective against a range of pest species, making it a broad spectrum activity. And nicotine is highly toxic. Its oral LD50 for rat is 30 mg per kg. However, it is rapidly detoxified and eliminated from the animal body. So in the presence of light, it decomposes easily. So nicotine is often used as the sulfate, representing 40% of the actual nicotine. It has a pyridine ring, so you have seen it before, and highly basic nitration on the pyridine ring is present. And the nicotine acetylcholine receptor, so here you can see the structure of the acetylcholine. Acetylcholine is a neurotransmitter in the central nervous system of insects, and nicotine is an agonist and mimics a part of the action of acetylcholine by interacting with the nicotine acetylcholine receptor. And they both seem to be structurally related, and comparison of the structural formula of the insecticidal nicotine and acetylcholine has been given here. So it has effects on nicotines on mammals. And here the metabolism, we can see the metabolism where the oxidation occurs on the fifth carbon compound. And nicotine is the active principle of the tobacco smoke, and as such, its metabolism has been studied extensively in man and animals. So oxidation occurs at the fifth carbon atom and the nitrogen atom and the N-methyl group of the nicotine to form uh, cotinine here. So here in the image, you can see the cotinine here. So methylation on the pyridyl nitrogen atom also occurs to give nicotine isomethonium. So different from animal metabolism, soil bacterium oxidizes nicotine on sixth carbon to form 6-hydroxynicotine. And in all animals tested, the major metabolic product was found to be cotinine, which is further converted to several metabolites and eventually to 3-pyridyl acetic acid. So these are the steps that nicotine follows, a pathway similar to that of non-nicotine. Coming to the therapy, nicotine affects the mammalian body, yes, and by stimulating the sympathetic nervous system, increasing heart rate and blood pressure, and leading to addictions. So, in the atomic nervous system, nicotine stimulates both sympathetic and parasympathetic pathways, which results in increased heart rate, BP, and gastrointestinal activity. In the central nervous system, nicotine enhances alertness, euphoria, and dopamine release, but may lead to anxiety and cognitive issues with chronic use. Coming to neuromuscular transmission, enhanced at low doses, but high doses or chronic use can lead to muscle weakness and paralysis due to receptor desensitization. Coming to the therapeutic approach to treat nicotine poisoning includes artificial respirations for as long as necessary until the body eliminates sufficient quantities of this drug. In addition, if the drug was swallowed, gastric lavage is helpful, and so is introduction of a slurry of activated charcoal into the stomach through a tube. So vomiting should be induced if the poison itself has not produced vomiting. Poisoning can also be treated with anticonvulsants. So this is how we treat if more nicotinic toxicity has happened. So that is the therapy here. And another compound is a pyrethrum, is one of the oldest organic insecticides known to man. Even today, it is preferred over the new generation insecticides for domestic use as it has low toxicity to mammals and high toxicity to insects, coupled with its remarkable effectiveness in causing insect knockdown. And it's basically taken from the dried and powdered flowers heads of the chrysanthemum. And the only species of commercial importance is chrysanthemum. It is applied to compounds having alcohol moiety similar to pyrethrins. The term pyrethroids refers to the insecticides based on the pyrethrin as prototypes. And pyrethrins are composed of an ester linkage between a cyclopropane alcohol, which is a cyclopropane ring where the alcohol will contains an hydroxyl group attached to it, and carboxylic acid, where the acid component usually consists of a carboxylic acid group, typically derived from the phenolic structure. With these groups being crucial for their insecticidal activity. So these two groups are very important for pyrethroid to act like an insecticide. So pyrethrins are esters formed by a combination of two acids basically, that is chrysanthemic acid and pyrethric acid, with three alcohols, namely pyrethroline, cineroline, and jasmolene. The esters of chrysanthemic acid are pyrethrin 1, cinerin 1, and jasmolene 1, and are together known as pyrethrin 1. The esters of pyrethric acid are pyrethrin 2, cinerin 2, and jasmolene 2, and are together known as pyrethrins 2. So these six components together account for the toxicity and knockdown properties of pyrethrum extract. So here you can see the copyrethrin biosynthetic pathway, like how it has been synthesized. Okay, and also the proposed model of the metabolic pathway, metabolism, like how it takes place here that you can see. So pyrethrins have a rapid knockdown action. The mode of action of pyrethrin is similar to the DTT, if not identical in both insects and mammals. And knockdown may be caused by hyperexcitation of the nerve, or nerve block is responsible for paralysis. Sublethal doses of pyrethrum spray force the cockroaches to emerge from their hiding places and run about frantically. So this is termed as a flushing effect. So it is much toxic to cockroaches in that sense. Coming to the metabolism, the terminal methoxy carbonyl of pyrethric acid moiety of the pyrethrin 2 is hydrolyzed. However, oxidation of the alcohol moiety is also important in limiting the toxicity of pyrethrins 1 and 2 here. The mode of action, as we have discussed, is like the knockdown method. A house-dust strain that is resistant to knockdown by DDT shows cross-resistance to pyrethrin. So the gene responsible for knockdown selectively confers resistance to the entire category of both DDT and pyrethroid compounds. And it is toxic to insects and mammals with the toxicity sublethal doses of pyrethrum spray, as we have seen in cockroaches, like with the flushing effect. And in the mammals, the acute oral LD50 of pyrethrins to rats have been variously reported of being 200 to 400 mg per kg body weight. Probably for the less pure preparations, maybe it can increase to 400 to 1500 mg per kg. So this is the acute oral LD50 value. And the dermal LD50 ranges from 1350 to 5000 mg per kg. And many humans may have skin problems from exposure to pyrethrins in the form of allergic reactions, which disappear rapidly on removal from exposure. So three major pyrethroid resistance mechanisms you can see in this slide. So that is at the target side, pyrethroid molecules, where they appear as gold spears here. They bind to the voltage-gated sodium channel to depolarize the motor neurons and cause paralysis. So this we can see here how it's causing the depolarization of motor neurons. And the target site mutations were subsequently selected in resistant populations. And in the B figure, now we can see the biochemical effectors which detoxify pyrethroids to non-toxic forms, how it's getting detoxified with the help of the CYPs. The excreted pyrethroid is indicated in gold spears. Here the CYP proteins are indicated in green and carboxylesterases are indicated in pink. And rejuvenation of CYP from blue oxidized to reduced green form can be seen here by nicotinamide adenine dinucleotide phosphate, NADPH, cytochrome P450 detectase. And in C, you can see the cuticular thickening or the modifications are also features of the resistance. So if the cuticle is thick, then it is difficult for the insecticide to enter into the cuticle of the mosquito. No? So that is what we are seeing here. Here you have seen the resistance and toxicity of cynarin 1 to insects is inferior to pyrethrin 1. But when synergized, cynarin 1 is as toxic as pyrethrin. Similarly, pyrethrin 2 and cynarin 2, when synergized, they become equally toxic, but to a degree around half to one third of the corresponding chrysanthemates. So, alethrin is not so synergized as compared with pyrethrins. So that is the toxicity. And another compound that is derived from the plants which acts as insecticides and which like everybody knows is the neem. Neem is a native to India and Burma. The neem tree is a member of the mahogany family. Also, it is known as the Margosa tree or Indian lilac. Azadirachta indica is its scientific name, and it contains a range of active compounds that acts as natural insecticides. So the primary bioactive compound responsible for neem's insecticidal property majorly is azadirachtin, though other compounds such as nimbin, selanin, and melantriol also contribute to its effectiveness. The main neem chemical is the mixture of several related compounds belonging to azadirachtin, melanotrol, salmonol, nimbin, and nimbidin, we will be seeing here. So azadirachtin has insecticidal properties, where the other nimbin has the pesticidal properties, and salanin affects the feeding behavior of insects. Nimbolide is a triterpenoid compound with insecticidal and antifungal properties. Margosine is an alkaloid compound which controls pests, and triterpenoids are a group of compounds with insecticidal effects. So there are also flavonoids that are present in the neem which acts as antioxidants and pesticidal activity along with the pesticidal activity. And coming to the essential oils, neem oil, it contains variety of fatty acids including oleic acid, linoleic acid, and palmitic acid, which contributes to its repellent and pesticidal effects. And Azadirachta is structurally somewhat similar to insect hormones ecdosons, and seems to be an ecdoson blocker so that insects do not molt. So the function of ecdysis we see there no yesterday. So that is similar to that of the azadirachtin compound here. It also interferes with the function to taste. Hence, many leaf-eating insects are deterred by plants to which even small amounts of azadirachtin have been applied. And it has effects on insects like neem preparation show the following effects like partial reduction or complete inhibition, reduction of the lifespan of the adults, oviposition repellents against females, and direct ovicidal effects can also be observed. Antipedant effects against larvae, nymphs, and adults can be seen, and formation of permanent larvae can also be seen with the azadirachtin growth. And growth regulating effects are molting between larval, instar, and especially in the prepupillar stage, and giving rise to larval pupilla can also be seen here. So neem acts on insects primarily by disrupting their molting, growth, feeding, and reproduction processes through the action of azadirachtin and other compounds. It is highly selective, causing minimal harm to humans, birds, and mammals, but it can have negative effect on non-target beneficial insects, aquatic life, and plants if used improperly. So while neem offers a more eco-friendly alternative to synthetic pesticides, its selectivity and dose must be carefully managed to avoid unintended ecological consequences. Neem and neem products are non-toxic to warm-blooded animals, though, and have been used as a medicine for generations in India. The seed kernels or whole fruits or leaves of Azadirach contain compounds high toxicity to mammals like milia atoxins. That is what about neem. So the other compound is retinol. It's a natural compound that is derived from the roots of several plants such as berries and logocarpus. So retinol occurs in certain leguminous plants. Retinol shows low toxicity to most of the mammals, but it's very much highly toxic to insects and fishes. And there are certain things that are involved due to the selectivity of its toxicity. We'll talk about it. And coming to the chemical structure, here we have seen the retinol. The structural formula of the retinol is seen here. Retinol is easily decomposed in the presence of sunlight and air and can therefore be used on edible produce just before harvesting only. And the photodecomposition of retinol led to at least 20 compounds when retinol was exposed to sunlight. So the optical isomers of the naturally occurring retinoids are L form, lever forms, and retinol content may be only a small percentage of the relative amounts in crude preparation. And the non-insecticidal toxicrol is often a major constituent of the preparation. As a main structural unit, every retinoid has a four-fused ring system that is chromaenochromanin from the parent heterocyclic system, which was in turn named from the parent heterocyclic system rotaxin. Rotenone is lethal to insects and fish, and its selectivity depends majorly due to the difference in most effective detoxification system that's present in mammals and protective barriers like blood-brain barrier and less efficient absorption of the compounds compared to insects and fishes. But however, retinol is severely toxic to insects and fishes because of their higher metabolic rates, less efficient detoxification, and vulnerability of their mitochondria to retinol-induced damage. Thus, while retinol is a potent pesticide for aquatic organisms and insects, it still poses less of the threat to mammals under normal exposure conditions due to these characteristics. So, coming to the metabolic pathway of the retinol here, you can see the steps and the process that are involved. So, initial metabolism of the retinol occurs mainly by oxidation of 8' 6 and 6, 7, and 12 positions on the methoxyl group as shown. And combination of these reactions gives many more metabolic products. All these metabolites are less inhibitory to the mitochondrial respiration than retinol. But 8' hydroxy retinol is as toxic as retinol to mice. So retinol metabolism occurs more rapidly with the microsome NADPH enzyme system than with other combinations of cofactors and individual fractions from homogenate of rat liver. Here comes absorption. The retinol is absorbed into the bloodstream and distributed throughout the body. Retinol is metabolized by CYP enzymes into hydroxylated and oxidized metabolites. And the formation of 6-beta hydroxyretinol, a major metabolite, can be seen here on the figure as well, on other hydroxylated metabolites. Formation of epoxide metabolites can be seen if epoxidation has happened, which are highly reactive and contribute to toxicity. And metabolites are conjugated with glucuronic acid or sulfates, making them water soluble, which we will see in the detoxification phase 2. And excretion, the conjugated metabolites are excreted primarily via urine or bile. The selective toxicity of retinol is closely linked to its oxidative metabolism and the differences in metabolic pathways between insects and mammals. Uh, as we have seen, they lack efficient antioxidant defense and have less effective detoxification mechanisms, making them more vulnerable to this compound. In contrast, mammals are better equipped to metabolize and detoxify retinol due to their antioxidant systems, blood-brain barrier, and cytochrome P450 enzymes, which can detoxify these insecticides, not making it more sensitive to mammals. This metabolic difference is a key factor that makes retinol more toxic to insects than mammals, thus contributing to its effectiveness as an insecticide. So in the fifth one, we have only these compounds that are derived from the plant-based products. So we have seen the nicotinoids, pyrethrins, neem, and retinoids are not only commercialized, but research on these plants has led to the development of new molecules, and each of these compounds has a unique mode of action. So we have seen in detail the nicotine. Nicotine, it is the principal alkaloid in the leaves of the nicotinate tabacum. And pyrethrum is one of the oldest organic insecticides known to man. And pyrethrins are esters formed by the combination of two acids, chrysanthemic acid and pyretric acid that we have seen here, which is along with the three alcohols named as uh, jasmolene. Pyrethrum has a rapid knockdown action, which we have covered in the fifth chapter. We have also seen the name, the Azadirachta indica, and the main name chemical is a mixture of related compounds and its role and its function. And we have seen the retinoids. So all these four bioinsecticides you can call because they have been derived from the plant sources. So retinoids are also a group of compounds in the roots of dairy species. And retinoid is lethal to insects and fish. It depresses respiration and heartbeat in insects, causing paralysis. So these are the compounds that you see, and some kinds of moral questions that you can expect from Unit 5R. What are the requirements of nicotinoid molecule to possess insecticidal activity? The different chemical ingredients present in neem and what are the effects of azadirachtin on insects? Or explain in detail the toxicity of nicotine to mammals and insects? Or what are pyrethrins? And explain with the help of structural formula the acid and alcohol groups involved. And also one more question that you can expect from this chapter is explain how retinol shows relatively low toxicity to most mammals, but very toxicity to insect and fish. Why it is like more toxic to insects and fish than the mammals that we have seen here in the present slide. So this is like a small chapter, and we are done with the fifth chapter, and we will be moving to the sixth chapter, that is organochlorines. So in this chapter, we see the chemistry, toxicity, and mode of action and metabolism of the organochlorines insecticides such as DDT and its analogs, lindane and its isomers, and the cyclodiones we'll be seeing in the organochlorines chapter. And organochlorines insecticides include a group of chlorinated hydrocarbons comprising of DDT and its analogs. And the presence of atoms of carbon, hydrogen, and chlorine combined within some cases of oxygen and including a number of uh, chloride atoms and bonds will be present. The absence of very reactive intramolecular sites can be seen in these organochlorines. And the presence of carbon ring structures, including benzene rings, is its property, and they're highly lipophilic, and the chemical instability in that they are not easily broken down in the environment. And we'll be seeing here DTD and its analogs, lindane and isomers. Here, even this is also a very small chapter, but we'll be able to finish this chapter here. So organochlorines are nothing but a group of synthetic chemical compounds that contain carbon, hydrogen, and chlorine atoms. So DDT and its analogs, so dichloro, diphenyl, trichloroethane. So we have been uh, hearing about this in every class because it is was synthesized and it was like very much potent back then in the olden days, which was later banned due to its harmful effects in the environment. So here you can see the chemical structure of DDT. It's a white, tasteless, almost odorless crystalline solid with needle-shaped crystals and a melting point of 108.5 degrees to 109 degrees. It is practically insoluble in water but soluble in many organic solvents such as acetone, benzene, carbon tetrachloride, kerosene, diesel, and so on. And it's slightly soluble in alcohol as well. So the commercial product usually contains about 70% of DDT, with the main contaminant being OPDDT, which is not as toxic to insects but may be more damaging to the vertebrate endocrine system. DDT is very effective against larvae of mosquitoes and black flies and against adults of all the important biting flies and against house flies, bed bugs, fleas, ticks, and other insects of public health importance. And here you can see the structure of the DDT. Uh, The DDT consists of trichloramethyl group CCl3 attached to a central ethane molecule that is connected to two phenyl groups, the benzene ring here. And coming to the DDT analogs here, the discovery of the insecticidal properties of DDT stimulated the search for analogs of organochlorine compounds. Although there are many compounds available, several analogs of DDT were in use earlier, but except dicofol, which is presently in use as an acaricide, none of them have been used like dicofol. Like it is like more effective than the other compounds that have been synthesized from the DDT. So here you can see the structure, the chemical structure of DDT and dicofol. So dicofol is a DDT analog that is structurally similar, but it only differs by the substitution of a chlorine atom on the phenyl ring with hydroxyl group attached to it. And dicofol also contains a chlorine atom in its structure, making it similar in terms of chemical properties to DDT. So here we'll see the comparison of DDT and dicofol. Coming to the structure, DDT is trichloromethyl group attached to the phenyl rings, while dicofol is similar to DDT but with a hydroxyl group present in it. And mode of action, it disrupts sodium ion channels in insect nerves, DDT, whereas dicofol disrupts sodium and potassium ion channels in insect nerves. And the target pests for DDT are it is a broad spectrum. It acts on mosquitoes and agricultural pests, whereas dicofol primarily it will act on mites and some insects. And DDT is highly persistent and it accumulates in the environment, where dicofol is persistent but somewhat less so than DDT. Coming to the toxicity to humans, moderate toxicity to humans, but at a potential carcinogen, this DDT. And dicofol is a lower toxicity to humans when compared to DDT, but yet still it is also toxic. Regulations, DDT is banned or restricted in many countries, but dicofol is restricted or phased out in some countries. Coming to bioaccumulation, it's highly bioaccumulating in food chains, and dicofol is bioaccumulating but generally less than DDT. And here, the toxicity of DDT increases with decrease in temperature. Hence, it is said to have a negative temperature dependence coefficient. And it also causes hyperextension of the legs and uncoordinated movements. Its poisoning causes this tremor throughout the body, loss of coordinated motion, that is ataxia, and hyperactivity resulting from external stimuli. Repeated falling on the back and writing efforts can also be seen. Disappearance of fast tremors can be seen. And finally, paralysis and death will also be caused due to the toxicity of this DDT. And coming to the toxicity in mammals, yeah, these are different theories. Toxicity in mammals, as we have discovered, yeah, moderate toxicity humans. Yet the acute oral LD50 of DDT is said to be 113 mg per kg of rats for rats, and a species which seems to be more susceptible to DDT than any other mammals. So coming to the mode of action, so chemically DDT is a comparatively inert molecule and hence has a residual problem and a magnification in food chains. The symptoms of DDT poisoning indicates that the site of action is the nervous system. So there are certain theories that we can see here. The blood toxin theory, where DDT enters the bloodstream, how it enters the bloodstream, and how it the mode of action, how it is done, can be seen by different theories here. We can see blood toxin theory, where the blood toxin theory was proposed by Sternberg and Kearns. According to them, the blood of DDT poisoned cockroaches contained a toxic factor other than the DDT. So this factor was toxic to flies and produced DDT-like effects in cockroaches' nerve cords. So later it has been shown by other workers that a variety of stresses, including electrical stimulations, non-specific stress, etc., produce similar toxic factors in cockroach blood. So this factor called autotoxin, is possibly released from the nerve as a result of hyperactivity of the nervous tissue. Hence, we can say in the blood toxin theory, the sterility enters the bloodstream, disrupting the nerve function. And another theory is dehydrochlorination. So Martin and Wayne suggested that hydrochloric acid was released at the site of the action by the dehydrochlorination of the CHCCl3 group, and that the nature of the ring substituents affected the insecticidal properties either because of electronic effect or because of solubility factors. However, insecticidal properties of DDT analogs which do not contain chlorine cannot be explained on the basis of this theory, that is dehydrochlorination theory. Coming to conductor 4 on toxophore group theory. So according to Logger et al., the role of CCl3 group was only that of a conductor 4 to bear the toxophore group to the site. So the combination in one molecule of both these moieties renders DDT an effective insecticide. Like the previous theories, this theory also fails to explain the insecticidal properties of the DDT analogs that do not contain chlorine, that is prolon, bulon, or dialin neopentane. So these are the DDT analogs that do not contain chlorine, so which we'll be not knowing about them, like how they are going to cause the toxicity or the mode of action. So these three theories can't explain them. And coming to the butterfly configuration theory, that was explained by Rogers et al. On the basis of work with DDT analogs. They proposed a theory that explained, among other things, the high activity of prolan. They consider the optimal stereochemical configuration of the DDT, where you can see in this picture, where the butterfly configuration of the DDT can be seen. It is seen like a butterfly here. So they further consider the CCl3 group as a bulk, which was important because it interacted with p-chlorophenyl groups to impart to the aryl rings the position of the maximum clearance and provide the butterfly configuration of the DDT. They supported their argument by citing the strong insecticidal activity of 1,1-bis methoxyprophenyl, 2,1-dimethylpropane, and the decreased activity of 1,1-bis p-methoxyphenyl methylpropane. But unfortunately, if one replaces CCl3 group of DDT by CHNO2Cl or CnO2Cl2, which are also bulk, the compounds are inactive. So that is the explanation with the butterfly configuration theory here. So the other theory, like the rotatability of benzene rings, has been further studied. So this theory relates the toxicity of such chemicals to the rotatability of the benzene rings about the alkyl carbon or a carbon to which they are attached. So from observations made on Stewart models, it was argued that when the oxophoric groups, that is groups present on the benzene rings, are attached in the RR prime position, the rotatability of the benzene ring is the greatest and hence the maximum. So insecticidal activity of DDT can be maximum and greatest during the rotation of the benzene rings, that has been told in the rotatability of the benzene ring here. So the objection to this theory is that in the case of DDT, the rings are also non-rotatable, but it is nearly as potent as insecticide as DDT. So coming to the metabolism of the DDT in insects, so there are 5 principal routes of metabolism in various organisms that can be seen while DDT. So here you can see the dechlorination of DDT to DDE. So the oxidation of the DDA will happen, and oxidation to dicophol will happen this in the second step, and oxidation to dichlorobenzophenone can be seen. Then dehydrochlorination to DDE, which is further reductive to dechlorination to DDT, can be seen here. And in metabolism in insects, if we talk in insects, the most important route of DDT metabolism is through its conversion to DDE. So DDT undergoes dehydrochlorination to form DDE, which is also toxic, and detoxification is often ineffective, leading to persistence and resistance in some insect populations. So the conversion of DDT to DDE is mediated by the enzyme DDT dehydrochlorinase, a glutathione-dependent enzyme, and the hydroxylation of DDT to dicophol is accomplished by the mixed functional oxidase system. So coming to the metabolism in mammals, so like most other species, mammals including man metabolize DDT to DDE, TDE, and DDA. How you can see here. Fine. So in mammals, DDT is metabolized in the liver into DDE and other metabolites like TDE and TDA. So mammals have more efficient detoxification and excretion systems, but bioaccumulation in fat stores can still cause long-term health effects in humans and mammals as well. And it has been seen that in blood, DTT and related metabolites are carried mostly by proteins, and less than 1% is carried by fat droplets in the blood. So DTT has been found in human milk in the range of 0.01 to 0.1 mg, and together with other metabolites, especially DDE, in ranges twice as high as that. In few countries, average values for total DDT in human milk of 1 to 5 mg per liter have also been reported. So conversion of DDT to dicophol occurs only in some insects. Methoxychlore is demethylated by the mixed function oxidase system to form the polar metabolites. So this is the metabolism of the DDT in insects and mammals. So coming to the linden. So linden in earlier literature was erroneously called benzene hexachloride. So how you can see here benzene hexachloride, where benzene along with three chloride molecules in the presence of sunlight, it gets converted to benzene hexachloride. However, if the chlorine atom is added to each double bond of the benzene so that the resultant compound is saturated, we obtain hexachlorocyclohexane, which we can see it down. So which is a derivative of the saturated compound cyclohexane, in which 6 hydrogen atoms have been replaced by 6 chlorine atoms. So this HCH, hexachlorocyclohexane, is prepared by the chlorination of benzene in ultraviolet light. The crude product is a grayish amorphous solid with a characteristic musty odor that is attributed to the presence of heptachlorocyclohexanes and other cyclohexanes. And here coming to the mode of action. So before the mode of action, we will just see the symptoms and the poisoning effects it does. So in case of mammals, linden stimulates the central nervous system, and as a consequence, the blood pressure rises and the rate of the heartbeat decreases. Bradycardia, which we call it as bradycardia. This is followed by increased rate of respiration, followed by restlessness and tremors of the whole body, salivation and grinding of the teeth, and finally convulsions. So this is the toxic effect that it causes in the mammals. And in man, the symptoms include dizziness, headaches, weakness, diarrhea, and epileptic form attacks can also be seen. The convulsive effects produced by linden are also antagonized by the alpha, beta, and D isomers of HCH. An oral LD50 for male rat of linden is seen to be 80 mg per kg. So these are the symptoms of the poisoning and the toxicity. So coming to the mode of action of linden, yes, linden acts by interfering with GABA receptor, gamma aminobutyric acid receptors, disrupting the chloride ion flow, leading to nervous system hyperexcitability, paralysis, and death in insects. Yesterday, we have seen the neurobiology of the insect, and we have studied the neurotransmitters, how they go, the chemical responses, the chemical synapses via the use of presynaptic and postsynaptic routes, and how they will interfere and how they cause paralysis and death. The same action will also happen in case of linden and other insecticides. The same mechanism happens. And coming to the mode of action here, the hypothesis is that linden antagonized the action of this compound at some vital site, and it is structurally somewhat similar to B vitamin myoinositol. Like here we can see the linden structure and the myoinositol structure, which are somewhat similar in structure. And the hypothesis that linden antagonized the action of this compound at some vital site was contradicted by the evidence no antidotal effects of myoinositol were observed against linden poisoning. Also, myoinositol is the isoster of 6-HCH, and hence this hypothesis does not seem plausible. Also, there is hardly any evidence to indicate that linden acts by interacting with a specific enzyme, though as compared to DDT, it is better inhibitor of the sodium potassium ATPase from American cockroach nerve cord. So linden and myoinositol share some structural similarities, particularly in their cyclic structures, but they have very different biological roles. And myoinositol is involved in normal cell signaling and insulin regulations, while linden disrupts neurotransmitter functions and is toxic to the nervous system. Thus, despite the structural similarities, their effects on biological systems are drastically different. So, one is crucial for normal cell functions, while other acts as a neurotoxic pesticide. So, in mammals, linden is metabolized by dehydrochlorination, hydroxylation, and conjugation to form several chlorinated phenols and conjugates, which are excreted in the urine. So here we can see the metabolic pathways and key enzymes that are involved and the metabolites they form upon the degradation and how they are excreted in insects. The oxidation, hydroxylation and conjugation takes place with the help of enzyme cytochrome P450 and glucuronidation. And the metabolites are hydroxylated metabolites 4-chlorocyclohexanol, which was excreted via urine, feces, but bioaccumulates in some fatty tissues, rendering more toxicity. While in mammals, including humans, so oxidation, dechlorination, and conjugation takes place. And the key enzyme process are cytochrome P450 enzymes, glucuronidation, and sulfation happens. And other key enzymes that are involved, the metabolism of 4-chlorocyclohexanol and dechlorinated products are the metabolites that are produced in mammals, including humans. The excretion is primarily through urine and feces. Whereas in rats, similar to mammals, more detailed research is available, and the key enzymes and the process that involves are again here cytochrome P450 enzymes and glucuronidation. So here in insects, mammals, and rats, we see the key enzyme process is CYPs and glucuronidation, and the metabolites are similar to that of the mammals, and it is excreted via urine, and lower bioaccumulation compared to the insects can be seen, whereas insects, the more bioaccumulation in the fatty tissues was observed. And coming to cyclodian insecticides. So cyclodian compounds are a group of highly active insecticides. However, many of them have long persistency and their use is restricted. So one of them is chlordane. The structure of the chlordane can be seen here. They include chlordane, heptachlor, aldrin, dieldrin, isodrin, endrin, uh, endosulfan, and the chlorinated terpene campechlor. So this is a group of highly chlorinated insecticides prepared by diene synthesis or Diels-Alder reaction. And in this reaction, a compound containing a double bond is added to the 1,4 positions of the conjugated systems. The hexachlorocyclohexane ring of chlorine has two possible configurations. These compounds are all cyclic, however, only a few of them are dienes. The exo and endo configurations can be seen in chlorines, and the reactions and dipole measurements indicate that it is in the form of endoisomers. Exo and endo configuration of chlorine describes the spatial arrangement of chlorine atoms around the cyclohexane ring. So here you can see the basic mechanism, a quick rule for discerning whether it's endo or exo. The endo configuration involves chlorine atoms positioned axially, that is inwards, while the exo configuration has chlorine atoms in the equatorial, that is outward position. The endo configuration tends to be more toxic due to the way it interacts with the biological system. It is insoluble in water but highly soluble in organic solvents. Chlordane is toxic to warm-blooded animals. Its acute oral LD50 for the rat will be between 457 to 590 mg per kg. Because it's easily absorbed through mammalian skin and skin contact should be avoided. In the presence of alkaline, chloridine loses HCl to form chemicals that are not insecticidal, and traces of iron catalyze this reaction. Steel containers therefore have a baked protective coating on the inside due to this reaction. Chloridine is used for household pests, pests of man and domestic animals, termite control, and also for agriculture and lawn pests. In agriculture, it is especially useful against grasshoppers and soil insects. So coming to the heptafluor. So heptafluor is a white crystalline substance with melting point of 95 to 96 degrees, and it is nearly insoluble in water but highly soluble in kerosene and aromatic hydrocarbons. So the technical grade material is a vaccine mass, and it contains about 70% heptafluor and 30% of the related compounds. So heptafluor is highly toxic, and its acute oral LD50 for male rat will be like 100 mg per kg, and it's easily absorbed through mammalian skin. And heptachlor is was used for the control of soil insects like wireworms, cutworms, and grubs. The epoxidation easily takes place in the treated animals and in soil. And in UV and sunlight, heptachlor is converted to photoheptachlor. And here you can see the chemical structure of the heptachlor, heptachlor epoxide, and photoheptachlor. So coming to the chemical structure of aldrin. So basically, these are all the compounds that come under cyclodiene insecticides. Okay, so one more is aldrin. So aldrin is also white crystalline solid with a melting point of 101 to 105 degrees and its vapor pressure at 25 degrees. The technical material contains not less than 95% of the aldrin and it melts at 45 to 60 degrees. So aldrin is almost insoluble in water but soluble in organic solvents. It's highly toxic and acute oral LD50 for rat will be like 55 mg per kg body weight and it is easily absorbed through the skin. So aldrin in contact with aldrin is a contact and stomach poison that also possesses a fumigant action. So it was used primarily to control soil insects, cotton rootworms, wireworms, white grubs, cotton insects, and turf insects. And coming to the structure of another cyclodiene, that is dieldrin. Uh, the dieldrin is an epoxide of aldrin. It is white crystalline structure, and the technical grade material contains not less than 85% of dieldrin, and it's a light brown material. It's highly toxic, and acute oral LD50 for rat being 46 mg per kg. And dieldrin is a contact and stomach poison that gives slow knockdown of treated insects. And dieldrin was also used for the control of cotton pests, grasshoppers, cutworms, and armyworms. The another one is the some metabolites of aldrin and dieldrin in living organisms. So you can see here some metabolites of aldrin dieldrin, where dieldrin uh, if an unidentified metabolites, if dieldrin gets converted to photodieldrin, then it gets to clean ketone or a pentachloroketone of dieldrin. And dieldrin can also convert to transdiol and it can form 9-syn hydroxydieldrin. So these are the some other metabolites. So aldrin and dieldrin can be converted to photoaldrin and photodieldrin in the presence of sunlight and also by microorganisms. So living organisms can also convert aldrin and photoaldrin to photodieldrin, which can thus be the terminal residue of aldrin and dieldrin. So photodieldrin is more toxic to aquatic and terrestrial organisms than aldrin or dieldrin. So the microsomal mixed function oxidase of rat, mice, and housefly, they metabolize photoaldrin at very low levels. But however, photoaldrin is converted in insects to clean ketone, which is even more toxic than its precursor. So coming to isodrine. So isodrine is a stereoisomer of aldrin. It's a white crystalline compound with a melting point of 240 to 242 degrees. Isodrine is highly toxic to mammals. The LD50 for rat is about 7 to 15 mg per kg, but it is no longer manufactured for use as an insecticide. Coming to endrine. So endrine is a stereoisomer of dieldrine, whereas you see isodrine is a stereoisomer of aldrine and endrine is a stereoisomer of dieldrine. It is white crystalline chemical which is insoluble in water and highly soluble in most organic solvents. And endrine is stable to alkali but undergoes rearrangement in an acid medium to an insecticidal inactive ketone. It's highly toxic and acute oral LD50 for rat being 7.5 mg to 43 mg per kg. So acute dermal LD50 for the same animal is 5.6 to 94 mg per kg. So the technical insecticide is brownish in color and contains not less than 85% of the active material. And engine was used for the control of cotton insects, cutworms, armyworms, plant bugs, and other pests. It is especially useful for the control of the black currant mite, which is naturally resistant to most insecticides and miticides. So Indian was also used for the control of pine mite in orchids. Because of its rearrangement to ketone in the presence of sunlight, it is not highly persistent. Yet it is highly toxic to fish, like 24 hours of LC50 for rainbow trout exposed to engine at 1.6 degrees was found to be 15 ppb. So the rearrangement products in the isodrine engine series, unlike photoaldrin and photodieldrin, tends to be less toxic than the parent compounds. And the other uh, ones in the cyclodienes are toxaphene and endosulfan, and these are its chemical structures. And toxaphene is one of the best known insecticides of this group, though it is not a cyclodine, but rather a chlorinated terpene. So it is prepared by chlorination of the bicyclic terpene camphene to a chlorine content of 67 to 69%. Mm, and its acute oral LD50 for rat is 69 mg per kg. It was used against all major pests of cotton and has low toxicity for bees. Whereas endosulfan here can also be regarded as a cyclic ester of sulfurous acid. And the compound is both an insecticide and acaricide. It is brownish crystalline solid, consists of two isomers, alpha and beta. So technical endosulfan is a mixture of these two forms, alpha beta, in the ratio of 4 is to 1. So it is insoluble in water but moderately soluble in organic solvents, and it is absorbed thoroughly. It is absorbed through the skin and very toxic to fish. Unlike most other cyclodine insecticides, it is low in persistency and still used. It undergoes oxidation in insects, mammals, and plants to form a primary insecticidal metabolite, endosulfane sulfate. So endosulfane and sulfate are hydrolyzed to form endosulfane diol. So endosulfane is effective for the control of beetles, cutworms, and aphids. It is highly toxic to fish yet. And toxicity and mode of action of cyclodiones can be seen here. So here the toxicity and mode of action of cyclodiones. As a group, the cyclodione compounds, like most other chlorinated hydrocarbon insecticides, act as a neurotoxicants. And the primary mode of action is by the blocking of GABA-gated chloride channels, but like HCH, we have said earlier, but not like DDT. So the important difference between DDT and the chlorinated cyclodiones is that the cyclodiones are absorbed from the intact skin. Uh, the difference between the oral and dermal LD50 values of the for the cyclodiones is much smaller than the difference for DDT. And coming to the mode of action, it is by the inhibition of GABA receptors and chloride channels. It causes excessive nerve firing and failure of nerve signal regulation, and it leads to hyperactivity, muscle tremors, convulsions, and paralysis. And acute toxicity is highly toxic to insects, causing rapid death upon exposure, and toxic to mammals and aquatic organisms like fish. Coming to chronic toxicity, bioaccumulation in fatty tissues leads to long-term exposure, and endocrine disruption, reproductive effects, and neurotoxicity can also be seen. Carcinogenity, some cyclodiones like dieldrin are probable carcinogens. And in insects, the cyclodiones have a positive temperature relationship, that is, their toxicity is enhanced as the external temperature rises. Whereas the opposite effect is seen with DDT and HCH. The symptoms of poisoning in insects resembles those of DDT, and the symptoms of poisoning in insect resembles those of DDT with strong tremors, short trains of impulses, but this effect appear 2 hours after treatment with dieldrin. And cyclodiones cause an excessive release of acetylcholine at presynaptic sites, but they do not inhibit the enzyme cholinesterase. So they do not affect any other enzyme systems. However, there is evidence that they interact with ATPases from nerve cord and muscles. With examples of houseflies and blowflies, a period of normal activity is followed by a more quiescent period, after which wing tremors become frequent, increasing in severity, and the insect shows spasms of wings beat without flying. So after a while, this culminates in uncoordinated flight, loss of ataxic gait, and failing on their backs. So this is how toxic it is to houseflies and blowflies. And this process is irreversible with so coming to the toxicity we are talking. So this process is irreversible with dieldrin and ends in death, but with related compounds that can be detoxified, recovery can occur even after 36 hours of immobilization, and the symptoms resembles those of an aesthetic effect. That is what happens in case of insects. And coming to the toxicity and mode of action of cyclodiones, especially in mammals, the most prominent symptoms of poisoning are convulsions. So these symptoms can appear even before any other signs of toxicity are apparent. Convulsions may be accompanied by confusion, excitability, incoordination, and in some cases, coma as well. So the toxicities of heptachlor, aldrin, and isoaldrin are increased due to their conversion to their corresponding epoxides, whereas heptachlor, epoxide, dieldrin, and endrin. So dieldrin is the most persistent among the cyclodiones, accumulating in fatty tissues and being retained for long periods of time, whereas endrin does not accumulate in fat and is metabolized to water-soluble metabolites, which are excreted. So most of the cyclodione compounds produce changes in liver cells, and some are considered carcinogenic to man. Because of their high oral and dermal toxicities, effects on liver cells, the potential for tumor production, and detrimental environmental effects, most of these insecticides have been banned from usage in agriculture and public health, except for termite control. So these are mostly banned for use by agriculture and public health, but however, they are being used in the termite control. So aldrin, dieldrin, heptachlor, and endrin undergo conversions in sunlight to form photoproducts. So these photoproducts are more toxic than their parent compounds. So whatever the photoproducts that are produced from aldrin, dieldrin, and heptachlor and endrin, so these tend to be more toxic upon exposure to sunlight. The photoproducts are formed, right? So they tend to be more toxic and more persistent in the environment than the parent compound. So that is what we mainly see here. So that is the toxicity of cyclodiones. Coming to the metabolism of the cyclodiones. So in earlier investigations on the metabolism of cyclodiones demonstrated the epoxidations of heptachlor, aldrin, and isodrin. So this is an activation process since the epoxides are more toxic than the parent compounds. So later, production of more hydrophilic and less toxic metabolites in insects and mammals was also reported. So these products are produced by hydrolytic processes or microsomal oxidation. The hydroxylated derivatives are either conjugated or excreted without conjugation. So in the case of dialdrin, at least 6 metabolites have been isolated in the urine and feces of rabbits, rats, and sheep. So two of these, namely, are trans 6,7 dihydroxy dihydroadrenaline, aldrin, mm, and 9 hydroxy 1, 2, 3, 4, 10 hexachlor 6, 7 epoxy octahydro octahydroendoxodimethanol naphthalene have also been identified. So, in addition, Matthews and Matsumura isolated and identified as a ketone oxidized dechlorinated metabolite that did not have an intact dialdrin ring system. So, the fate of aldrin 614 in maize, wheat, and soils showed the main radioactive products to be dialdrin and dihydrochlorine dicarboxylic acid. So trace amounts of aldrin, photodialdrin, and unknown acidic compounds and unknown polar metabolites were also formed. So the metabolism of endosulfan is also complicated. It forms endosulfan alcohols by hydrolysis and it oxidizes to endosulfan sulfate. The hydrophilic metabolites are also formed in the case of endosulfan metabolism. Though a endosulfan decomposes fairly rapidly, like 50% approximately within 60 days, with the formation of endosulfan sulfate, which was found to be relatively more stable. Beta endosulfan was found to be much more persistent, that is, approximately 50% in 800 days can be seen here as compared to the other compound. So it is the decomposition, how faster the decomposition happens. So alpha endosulfan can decompose fairly rapidly compared to the beta endosulfan, where alpha endosulfan can take approximately 60 days to degrade to 50% of it, whereas beta endosulfan it takes around 800 days to decompose to 50%. So you can see that it is much more persistent than alpha endosulfan. So Mirex is relatively very stable and is not easily metabolized by animals. Its photodecomposition is slow. In mammals, isodrin is epoxidized to endrin, which is oxidized without skeletal rearrangement and excreted. So that is the metabolism of cyclodione. Here in insects, the metabolic pathways are hydroxylation takes place, and the key enzyme is cytochrome P450, and the end product metabolites are hydroxylated metabolites. And also dehydrochlorination takes place. The dechlorinated metabolites will be there, and epoxidation also takes place in insects. Coming to the mammals, the oxidation through cytochrome P450 takes place by the action of the enzymes, giving rise to hydroxylated metabolites like dialdrin. Dehydrochlorination takes place, resulting in the conjugated metabolites glucuronide, and conjugation with glucuronic acid takes place with excreted metabolites in the form of urine and feces. And coming into the birds, the hydroxylation and epoxidation takes place with the help of the key enzymes, cytochrome P450 enzymes, the CYP enzymes, and hydroxylated metabolites are the end products of it. Whereas in dehydrochlorination, dechlorinated metabolites can also be seen. So thank you. So this is about the uh, these two chapters that we have seen here, the organochlorines, and also we have seen the plant-based botanical insecticides, where we have studied the nicotinoids, their physical and chemical properties, the structure-toxicity relationship, and their mode of action, and effects of nicotine on mammals and their action. And we have also studied the organochlorine in fifth and sixth chapters here. In the coming class, we'll be seeing the organophosphorus insecticides, um, where we'll deal with the its introduction, structure, and nomenclature, and symptoms of and their poisoning as well. So there are many isomers of HCH, of which seven are alpha, beta, gamma, and so on. Cyclodine insecticides includes chlordane, heptachlor, aldrin, dialdrin, and isodrin. So this is about this two chapters that we have finished. Thank you.